Hello everyone, welcome back to my course on understanding the brain. Today we are analyzing the synapse, their structure and how they function, because it is a crucial topic, because understanding the fundamental logic behind the synaptic activity allows us to apply this knowledge to various complex neural systems. Let's start. So we are discussing today the key event occurring at synapses, and for that we will focus specifically on chemical synapses, which rely on molecules to transmit signals from one cell to another. But before we begin, we will outline our main objectives. So by the end of this course, we should be able to understand the basic structure of synapses and to be able to explain the process of synaptic transmission. So here is the synapse, a representation of the synapse. It is basically a connection, a specialized junction between two cells. It can be two neurons, for example. It's basically one cell talking to another. At the synapse level, the information is transferred and it takes place between a neuron and another neuron, or another muscle, or another gland cell. So here we have two neurons. We can recognize the neuron based on the soma and the specific neurites or branches or processes, the dendrites. As we remember from our last course, the diameter of the dendrites are getting smaller at the end. So here are the dendrites, the nucleus, the soma, and we can recognize the axon, this long fiber, and with the yellow flash we can identify the action potential or the electric impulse that are running through the axon. Here in this picture, this specific axon has three collaterals and it connects with the next neuron. It is a neuron based on the dendrites, the specific dendrites, specific branches that are represented here. So what is the synapse? The synapse is this connection junction between one neuron with another neuron. We can zoom in here and we can see the actual connection or synapse. And the synapse is structured, it has at least three main components: the presynaptic neuron, terminal, the postsynaptic neuron receptor, and the synaptic cleft where the information is transmitted. We will explain this figure later on. So as we can see in this picture, the connection can be between the axon and the dendrites. Those are the most common synapses in the nervous system. We also can have a connection between the axon and the soma. It will be as an axon somatic synapse, and also we can have other types of synapses. We will mention them later on. Some fun facts about the synapses. The average human brain has around 100 trillion synapses. That's more than the number of stars in the Milky Way galaxy. During the development, the 100 trillion synapses in the human cortex formed at a rate of an estimated 10,000 every 15 minutes. To be able to remember what a synapse is or the structure of a synapse, we can make several types of comparison. We can make an analogy with the post office. The presynaptic neuron is like the sender, the neurotransmitter are the letters, the information would be transmitted on the letter, and we have the postsynaptic neuron, which will be the recipient. This can help us make this abstract concept more relatable. We can also discuss about synapses in terms of communication networks, just like how computers send data through networks, and our brain also sends signals through our synapses. We can also compare the synapse with a bridge, and we can use the bridge metaphor. It is like the neurotransmitter is like a car that crosses the bridge. The bridge is the synaptic cleft, and it crosses the bridge from one side of the presynaptic neuron to the other side, postsynaptic neuron. Some synapses can transmit signals in just a fraction of a millisecond. It is very, very fast. And also our brain forms new synapses whenever we are learning something new. I hope you'll learn something new today during this presentation, and you'll build up new synapses. So our brain is literally building itself as we learn. So here are the structure of the synapse. In the beginning of the lecture, I told that today we are discussing the chemical synapses, but there are, just for you to know, that there are also electrical synapses where action potentials jump directly to the next neuron, but these are rare and they are more characteristic for invertebrates. In our brain, we can assume that for the most part only chemical synapses function. Of course, we have electrical synapses as well, and primarily we can find them in areas like the retina. But today we are discussing the structure of the chemical synapse. The chemical synapse will use a molecule to transmit the information. So here we can see a nerve cell at the end of the axon, or the axon terminal. We can call it the axon terminal, and based on the synapse, or discussing the synapse, we will call it the presynaptic terminal. Is the part responsible for transmitting signals. The action potential, so the brief electrical impulse, will travel along the axon and will arrive here at the axon terminal. Here we have those are very important elements of the synapse. Here we have vesicles that they contain this chemical mediator or the neurotransmitter. Those are called also the synaptic vesicle. This synaptic vesicle will bind to the presynaptic membrane, and then they will diffuse or will throw the neurotransmitter in the synaptic cleft, in the narrow space between those two neurons. in this picture, for the didactic purposes, this synaptic cleft is drawn a little bit with space. The space is not that narrow. Usually, or in reality, the space is quite narrow, but there is a space between those two neurons. The space is needed for the fact that if it won't be a space, the electrical impulse will jump directly to the next neuron, and some molecular mechanism that will mediate the transmission of information won't be possible. That's why we have this space, this gap, this synaptic cleft between those two neurons. So just to come back, the neurotransmitters are released in the synaptic cleft, and the next phase is that they will bind to the membrane of the postsynaptic membrane of the postsynaptic neuron. Here at the surface of the membrane, it can be a dendrite, we have the receptor. The receptor that will capture the neurotransmitter, and the neurotransmitter then will generate an electrical impulse that will run over the dendrites to the soma, and from the soma to the axon, and from the axon to the next cell, and so on and so forth. So just to not just to sum up, but to retake all the information, we have the presynaptic terminal or the axon terminal with vesicles filled with neurotransmitter. Of course, here we have other organelles represented in the picture, like mitochondria. We also have calcium ion channel, because we will see later on or in other lectures that for the movement of the neurotransmitter or of the vesicles and the binding to the membrane, we need calcium, and we have this calcium ion channel that will, when the action potential will arrive to the end of the terminal, they will generate or trigger this ion channel to open. In the outside space of the cell, there are a lot of the concentration of the calcium is higher than inside the cell. This will trigger the fact that the calcium will enter the cell, and the calcium will bind to the vesicles, and they will make the vesicles bind to the membrane, and in the membrane will the vesicle will release the neurotransmitter in the synaptic cleft. Here at the postsynaptic membrane, we have the receptors. The receptors are proteins that will detect and will respond to neurotransmitter. So every single element discussed here or mentioned here are important for the synaptic transmission, and basically form the synapse and make the synapse function. Some data, some facts about the scale of the synapse, just to make an idea of the sizes of the synapse. A synaptic vesicle is around it's around 20, 30, 40, even 60 nanometers wide, which is and the synaptic cleft is about 20, 30 nanometers wide. So as we can see, the vesicle have roughly the same size as the cleft, so the gap is quite narrow. It's very close to the diameter of the vesicle, so this spaces ensures that neurotransmitter will diffuse quickly, but also the synaptic cleft is wide enough to prevent direct electrical transmission. This allows for complex signal processing, and we will see that in our future courses. I started the discussion of the types of synapses when I defined the synapse. So in this picture we have represented some types of synapses, and we have the axosomatic synapses. Axosomatic synapses are represented here. It's the connection between an axon and the soma. We also have the extracellular connection, when the axon with no connection secretes into extracellular fluid. We also can have axodendritic connections, synapses, when the axoterminal ends on a dendrite spine. Here we can see represented this axodendritic synapse. Also, we can have an axoaxonic synapse when the axon terminal secretes into another axon, and we have represented this type of synapse in here. And also we have axosecretory synapses when the axon terminal secretes directly into bloodstream. So here are the capillary, represented the capillary. So basically we have a variety of the synapses, and in densely packed neural areas such as the thalamus, for example, a variety of synaptic arrangement exists. Synapses are typically central synapses occurring in the brain or spinal cord. However, there are also neuromuscular synapses and autonomic synapses, where the postsynaptic cell may be a muscle cell, a glandular cell, or another organ cell. So in the previous picture we had a representation of the synaptic, a drawing of the synapse here, and I mentioned the source on my slide. There is an image of an actual synapse. It is made with a with a microscope, an electronic microscope, and we can see represented here the synapse. So with the red arrow there is a synapse here, and those black dots are the vesicles that are coming to the terminal and will diffuse in the synaptic cleft. And with the green arrow it is represented another synapse. Here I mentioned again the synaptic transmission process, and we have 5 stages of the synaptic transmission. The first stage is the arrival of the action potential. An action potential travels along the presynaptic membrane. The second stage is vesicle movement. The action potential will trigger the vesicle movement towards the membrane, the presynaptic membrane. The third phase is the neurotransmitter release. Vesicle will fuse with the presynaptic membrane and will release neurotransmitter in the synaptic cleft. The fourth stage is the neurotransmitter diffusion. The molecules will travel across the cleft and will bind to postsynaptic receptor. And the last phase is the postsynaptic response. If the synapse is excitatory, neurotransmitter binding will lead to sodium influx and will trigger a new action potential to the next cell. If the signal is transmitted successfully, the information will continue to travel through the nervous system, potentially triggering responses or being stored as a memory. If the synapse is inhibitory, the information will be inhibited and not transmitted to the next cell. Some facts about the synapse size and variability. So the central synapses, or the synapses that we can find in the brain or spinal cord, are usually 1 to 2 micrometers in diameter. The peripheral synapses, such as neuromuscular synapses, here in the picture we have represented this neuromuscular junction, are much larger, sometimes between 10 to 100 times bigger than central synapses. And the largest synapses are neuromuscular synapses. They use acetylcholine as a neurotransmitter. Okay, we know that the neurotransmitter is the key to the chemical synapse, so we will discuss here on this slide the life cycle of the neurotransmitter. So we know that here in the end of the terminal there are vesicles with the neurotransmitters, and they will be released and diffused in the synaptic cleft. But how many vesicles with neurotransmitters are? Are they ending? At what point they are made? Who is making them? So we will discuss that here. So of course there is the life cycle of neurotransmitter consists of 4 stages, and the first stage is the synthesis and storage of the neurotransmitter. So the neurotransmitter must first be produced and stored in vesicles, and then accumulated in the presynaptic terminals. So the first stage is the synthesis. It can be synthesized in the soma next to endoplasmatic reticulum and the mitochondria, because we need also energy to produce and synthesize the neurotransmitters and package them into vesicles. And we can synthesize them. The second way is to synthesize them here next to the axon terminal. The second stage is the release, and when an action potential will arrive, the neurotransmitter will be released into the synaptic cleft. Also we have the third phase, receptor interaction. The neurotransmitter will bind to the receptor on the postsynaptic membrane, which can be either the reaction will be either to excite or to inhibit the next cell. And the last phase is the inactivation. Once the neurotransmitter has acted on the receptor, it must be removed to prevent prolonged signaling. So to to illustrate this, we can consider the neurotransmitter as a finger and the receptor as a button. So when the neurotransmitter is released, the finger will press the button and the button will have a reaction, will activate something. Of course, if the neurotransmitter is not deactivated, this button will be pressed for too long. So it will get stuck like a keyboard key that doesn't release properly, and this sticking effect must be avoided to ensure that the signal's transmission has a clear beginning and a clear end. So for that purpose, there are special molecular mechanisms that exist, and they remove the neurotransmitter generally or often. Those mechanisms involves enzymes that will break down those neurotransmitters. So that's basically, in a few words, the life cycle of the neurotransmitter. Okay, so the neurotransmitter is synthesized, as I said, either in the cell body of a neuron or directly in the presynaptic terminals. We have some elements of neurotransmitter synthesis. We need a precursor molecule, or sometimes multiple precursor, a specific enzyme that will facilitate the reaction of forming a new molecule from a precursor molecule, and also we need ATP, we need energy to drive this process. The enzyme is a molecular machine that will capture the precursor molecule or molecules. It will change the shape and combine them into a neurotransmitter, and it will release the new form neurotransmitter, will reset, and will repeat the cycle. So basically, the synthesis of the neurotransmitter is regulated like this. So the first method is considered more ancient and is common to all cells, and it makes sense to synthesize the neurotransmitter next to the organoid specialized for the synthesis of the molecules. Since protein synthesis occurs near the nucleus, so it is efficient to produce neurotransmitter here. Also, there is another way of synthesis of the neurotransmitter here at the terminal, and an example of a synthesis of a neurotransmitter that will be synthesized here is the acetylcholine. It is known for its role at the neuromuscular junction, the synapse between a motor neuron and the muscle fiber. So in the presynaptic terminal, acetylcholine is synthesized from acetyl coenzyme A, acetyl-CoA, and choline via the enzyme choline acetyltransferase. So the level of enzyme activity is the rate limiting step in the synthesis pathway. The acetylcholine will be packed in vesicles and for storage in the terminal, and it is packed via the vesicular acetylcholine transporter. So as we can see, there are two ways or two places where the neurotransmitter is produced. So here we have represented the way of the first way of the synthesis of the neurotransmitter. Why the first way is not very efficient? Because like other cells, neuron cells have this long process, and sometimes it takes time to transport the vesicles with the neurotransmitters to the terminal. So that's why also for some neurotransmitter it is more efficient to produce them here at the axon terminal. In order to transport the neurotransmitter to the terminal, neurons are using microtubules. Those are tiny protein, and they are like highways built from tubulin, and they help transporting the vesicles to the terminal. Of course, it takes times. Some peptide neurotransmitters are synthesized in the cell body because they use specific precursors that are produced here at the endoplasmatic reticulum. Smaller neurotransmitters are produced in the terminal. This local production allows neurons to quickly replenish neurotransmitter stores and adapt to the demand. Okay, and there is also some information about the vesicle loading and recycle, how the vesicles will load and recycle. So the neurotransmitter will arrive, it will be synthesized, of course, will arrive, will be loaded in the vesicles. The vesicles will bind to the membrane, presynaptic membrane, will release the neurotransmitters in the synaptic cleft, so it will become empty. So, and this will be reused for the cycle will be retaken to refill again the vesicles. So of course it's an ecological efficient way of reusing these vesicles, and to not to produce a lot of waste in the body. And how the neurotransmitter release is triggered, we will discuss briefly this process. I mentioned a little bit in the first slide. So when an action potential will reach the presynaptic terminal here, it's not very, we cannot see very clear, but before this axon terminal we have this voltage gate calcium channel. So these channels will allow calcium ions to enter the cell. So inside the cell and outside the cell, the concentrations of the calcium ions is different. So outside the cell we have 10 times or even 100 times, 1000 times more calcium outside the cell. And when the electrical impulse or the action potential will reach the presynaptic terminal, it will activate those gates and the calcium will enter the cell. The calcium influx will trigger the vesicle fusion, and it will lead eventually to the neurotransmitter release. So from this point, the neurotransmitter will interact with the receptor on the postsynaptic membrane, and the process of transmitting the information will continue. So basically, I hope I covered enough on the synapse and the synaptic structure and the synaptic, a little bit of the synaptic function. And now, to conclude, I will retake the three key component of the synapse: the presynaptic neuron, the synaptic cleft, and the postsynaptic neuron. The key structure that helps the communication between the two neurons or the two cells are the axon terminal, the synaptic vesicles filled with neurotransmitters, and the receptors. We have two types of synaptic: electrical and chemical. We covered today the chemical synapse, and we discussed about the neurotransmitter synthesis and the vesicle loading and recycle. Some key elements, of course, things are a lot more complicated than discussed here, but for the basic for a basic understanding, I hope those information are enough for understanding. I hope you would learn something new here, and if you need to retake and rewatch some parts of the videos, I invite you to do so. So I would love to hear also your thoughts in the comment, and feel free to check out my other videos for more topics. See you next time, and thank you for watching.